This is the Phoenix crystallization robot. It's made by Art Robbins Instruments, and it's a microfluidics robot designed specifically for setting up crystallography screens. The 96 head is responsible for moving the well solutions that will actually be screened in these experiments, and the nano needle is responsible for distributing proteins to the tray. There are also wash stations for each head, and racks that slide out to hold sample trays, deep well trays with well solutions, and a cooling rack for holding your protein sample. The entire system is controlled through software on this computer, and there are pumps responsible for moving fluids through the system. These pumps control the protein and well solutions moving through the heads, and these pumps pump water to the wash wells and out again to wash the heads. There are 3 things that I need to run the most basic program that I use, which is setting up a single protein with 96 conditions. The first of them is a sample tray, which will hold my proteins and well solutions while crystals are hopefully growing. And it's set up in a 96 well format. Each reservoir actually contains 3 small sample wells and a deeper well, which will hold the crystallization solution that we're testing. This tray gets loaded on the top rack for this program, and the A1 is lined up with this corner. The next thing that we need is the actual well solutions that I'll be testing. Again, it's in a 96 well format, and usually this is a kit that we purchase, and sometimes we'll make dilutions of that kit so that we can test them as a slightly different condition. And also you can set these up by hand if you're trying to optimize a condition that you may have found from a previous screen. So again, this is lined up on this shelf with A1 coordinated with where A1 will be on the sample tray. And the last thing that we need is our protein sample. And you can use as little as 30 microliters with this robot, which is very helpful if you have a protein like mine that you can't purify very much of at a time. And you only use about 19 microliters total at the smallest drop size that we can use, which is 0.2 microliters of protein and 0.2 microliters of well solution. And that also goes here. This software allows us to optimize the program that we want to use. We can change the volume of the drop size, we can change the well solution, how viscous the well solution or the protein solution may be, and also washes, which are very important for these tiny needles that we're using. For this program, I'll be doing the smallest drop size, which is 0.2 microliters. And if you can think about pipetting that repeatedly by hand, you probably wouldn't want to. And the first thing that you'll see happen is a wash, followed by dispensing of the well solutions into both the deep reservoirs and the sample reservoirs. And then the protein will get dispensed. It will give me the tray so that I can seal it, and it will wash again at the end. So we go ahead and start it. So this reservoir just filled with water, and now the needles are pulling up water to rinse themselves. Before doing this run, I did a full wash of both heads. So this reservoir is now also filling with water. And the first step is to push air out of the needle and then to draw air up to wash the needle and the tube. And it's important that we see vigorous bubbling during this washing so that we know that there aren't any blockages in the needle. So now the 96 head will pick up 50 microliters of our well solutions from this deep well block. And it gives it a second in case those solutions are very viscous so that they can all draw up to the same volume. So now it's dispensing 40 microliters of each of those well solutions, and it will mix them to make sure that they're spread evenly around the reservoir. and it just added 0.2 microliters of each of those well solutions to the small sample reservoirs next to each deep reservoir. Now the nanoneedle will pull up 25 microliters from our protein tube and dispense 0.2 microliters into each of the same small sample reservoirs. And you'll see that it works by moving the sample rack back and forth rather than the nanoneedle. And the nanoneedle is timed to very precisely dispense exactly when it's over those tiny sample wells. So now it presents me with the tray so that I can seal it before those drops start to evaporate. After removing the sample tray from the rack, we need to seal it quickly before those tiny drops evaporate. We do that with special, very clear tape. And this tape is also less likely than others to form condensation on its surface. And then we seal each reservoir carefully by smoothing them over. Trim them. And then they can get stored in boxes like these that are protected in this room, which is temperature controlled. And also the boxes are stored on foam to limit vibrations that the samples will feel. And then they get checked periodically until crystals grow.